Hi, my name is Allie, and my partner's name is Julia, and we will be telling you about lysosomes and peroxisomes.
So beginning with lysosomes, the word lysosome itself has the prefix lys, which can be defined as to cut or digest, and this makes sense because the primary function of a lysosome is digestion and waste removal. With their digestive hydrolytic enzymes, they digest waste, organelles that are in excess or worn out, food particles, and bacteria. It then recycles all of this. They are only found in the membrane of an animal or eukaryotic cell, and as you can see in the picture on the right, they are formed in the endoplasmic reticulum and move to the Golgi apparatus on the transport vesicle. Then the lysosomes move to the cytosol. In the picture to the left, you can see a lysosome structure, and the center is the enzyme complexes, and it is surrounded by a membrane that is made up of phospholipids. In general, lysosomes range from 0.1 to 1.2 micrometers in size.
Now I will tell you about phagocytosis and autophagy. So if there is something in the cell that is no longer functioning, then the lysosomes will digest the organelle that is no longer helping out. An example of this could be if a mitochondria has been worn out. This is the process of phagocytosis. Another example of this is if a white blood cell absorbs a harmful bacteria, then the lysosomes will absorb the blood cell and release the hydrolytic enzymes that are inside, which will then destroy the bacteria from the white blood cell. So as you can see in the photo, if something enters the cell, such as food, then the lysosome will attach itself to it and use the digestive enzymes to digest this. And so after they break down everything into the most basic building blocks, they will release it into the cytoplasm so they can be reused again. An example of this could be if there are extra hormones in the cell, then the lysosome will break that down into the proteins, and then from there the amino acids will be released. So autophagy is the process of eating yourself, and they use their enzymes to recycle the cell's organic material. And the picture to the right you can see was taken in a microscope, and where the arrow is pointing, that is a lysosome. Now I will tell you about apoptosis. And so apoptosis refers to the process of when the hydrolytic enzymes are held together by the membrane, but then that membrane bursts within the cell. So the enzymes inside the lysosomes can only function when they are in an acidic environment. And the pH inside of a lysosome is around 5. But if the lysosome breaks, the enzymes inside will not do anything since the pH of the cytoplasm is around 7.4. So as you can see in the photo to the bottom right, if there's a disruption in the membrane, then the lysosomes and the enzymes are then free. But if multiple lysosomes break at the same time, that will be enough to lower the pH, and therefore the enzymes will be able to activate, destroying the cell. This is the process of apoptosis. Now I will tell you about peroxisomes. So as you can see, this photo was taken in a microscope, and the peroxisomes form the shape of a ball. They closely resemble lysosomes because they also have various metabolic functions, and they are most commonly found in eukaryotic cells. Their main function is to rid the body of toxic substances, which will now be further discussed in the next slides.
So this is just an overview of the peroxisome organelle, and the lipid bilayer membrane surrounds the crystalline core, and this is what controls what enters the peroxisome, and then the crystalline core in the center executes the functions of the peroxisome inside the organelle. So the main role of a peroxisome organelle is to protect the cell from damaging effects of hydrogen peroxide, and it does this by isolating hydrogen peroxide, and with its enzyme called catalase, it turns the hydrogen peroxide into water and oxygen. This can be shown in the picture to the left, which shows the reaction equation. So it's H2O2 hydrogen peroxide with the enzyme catalase then turning into water and oxygen. This functions to also detoxify alcohol and other harmful compounds by removing the hydrogen atoms and transferring them to oxygen atoms to become hydrogen peroxide, which the peroxisome organelle can then turn into water and oxygen. In the picture to the right, you can see the peroxisome organelle inside a cell. So, in conclusion, peroxisomes and lysosomes are vital for the cell because of their useful metabolic functions.
Thank you for listening.